Today we are going to talk about gastrulation, otherwise known as the development of the trilaminar germ disc. Let us begin by watching this very short video animation on gastrulation.
By the end of the second week of development, the bilaminar embryonic disc, consisting of the hypoblast and epiblast, has formed. Throughout the third week of development, this bilaminar disc differentiates to establish three primary germ layers, in a process known as gastrulation. Around 15 days after fertilization, a thickened structure forms along the midline in the epiblast near the caudal end of the bilaminar embryonic disc. This is called the primitive streak. At this stage, the formation of the primitive streak defines the major body axes of the embryo, including the cranial end towards the head and caudal ends towards the tail, as well as the left and right sides of the embryo. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node which contains a circular depression, known as a primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak, forming a primitive groove. Once formed, cells of the epiblast migrate inwards towards the streak, detach from the epiblast, and slip beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate through the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are eventually completely replaced by a new proximal cell layer, which is referred to as the definitive endoderm. By day 16, the majority of the hypoblast has been replaced. The remaining cells of the epiblast are now referred to as the ectoderm and forms the most exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form a germ layer known as the mesoderm. Once the formation of the definitive endoderm and mesoderm are complete, epiblast cells no longer migrate towards the primitive streak. Throughout gastrulation, the ectoderm continues to form from the cranial to the caudal end of the embryo, establishing three distinct primary germ layers throughout the whole embryonic disc. The gastrulation process is finally complete.
One of the most important events that happens during gastrulation or the third week of development is the formation of the three germ layers, the ectoderm, mesoderm, and the endoderm. Gastrulation begins with the appearance or the formation of the primitive streak. At first, the primitive streak is not very defined, but eventually in a 15-day-old or a 16-day-old embryo, the primitive streak will be clearer and its structural parts would also be more developed, such as the presence of a primitive groove and also a depression called the primitive pit. So this is a 15 to 16-day-old embryo with a very clear primitive streak. As soon as the primitive groove appears, cells from the epiblast will begin to migrate towards the primitive streak. And when they arrive at the primitive streak, the cells will change their shape. They will separate from the rest of the epiblast and will move inward. This inward movement is called invagination. Now, the reason why cells are migrating is because of the presence of the gene fibroblast growth factor 8. This FGF8, which is synthesized by the primitive streak itself, downregulates the expression of E-cadherin. The protein E-cadherin is the one responsible for keeping the cells in the epiblast together or adhere to each other. But because it is downregulated, they lose their normal attachment to their neighboring cells. Another important thing that happens here is that as the epiblasts migrate, some of these epiblastic cells will displace the cells found in the hypoblast until eventually all of the cells in the hypoblast are completely replaced. Now, the hypoblasts that have been replaced will become the future endoderm. In the meantime, some of the migratory cells will move in between the future endoderm and the epiblast and will remain there. That would be the future mesoderm that is shown in red in this figure. On the other hand, some of the cells will remain in the epiblast, and these will become the future ectodermal cells. In the cephalic region, take a look at figure A here. Some of the migrating cells will pass on each side of the precaudal plate. The precaudal plate will later form between the tip of the notochord and the oropharyngeal membrane and is actually derived from some of the very first cells that would migrate through the node in the midline. These cells will move in the cephalic direction, and later the precaudal plate will become very important in the formation of the forebrain. Meanwhile, the oropharyngeal membrane that you see there at the cranial end, it's really just a very small region made up of ectoderm and endodermal cells that will become the future opening of the oral cavity. The cloacal membrane that you see at the caudal end will become the future opening of your anus. The formation of the notochord involves morphogenetic movements such as invagination, which is the moving inward of cells, and intercalation, which simply means the insertion of cells to a different layer. Let's begin with the invagination of prenotochordal cells in the primitive node. So the prenotochordal cells invaginate in the primitive node will move forward cranially until it reaches the precaudal plate. The precaudal plate, notice, is just caudal to the oropharyngeal membrane here. The oropharyngeal membrane is made up of ectoderm as well as endodermal cells. There are no mesodermal cells making up the oropharyngeal membrane. The same thing is true for the cloacal plate or the cloacal membrane, which is also made up of tightly adherent ectodermal and endodermal cells. Now, the oropharyngeal membrane, as we mentioned earlier, will become the future opening of the oral cavity, while the cloacal membrane will become the future anus. So what happens to the precordial cells? The precordial cells will eventually insert itself here in the hypoblast layer, which is eventually replaced by cells coming from the epiblast through intercalation. So the pre-notochordal cells become intercalated in the hypoblast, and eventually it forms what you call as the notochordal plate. Now take a look at the point where the primitive pit forms an indentation in the epiblast. That would be here. So see that indentation? That's your neurenteric canal that would temporarily connect the amniotic cavity with that of the yolk sac cavity. With the formation of the cloacal membrane at the caudal end of the embryonic disc, an extension of the yolk sac would be formed, and that extension would be the allantois that you can see here. This structure is actually vestigial or rudimentary in humans, but for other lower forms of vertebrates, it serves a function for the secretion or excretion of waste materials. Now, while it is rudimentary for man, apparently, if it remains, it may be involved in abnormalities of bladder development. Finally, as the hypoblast is replaced by endoderm cells moving in the primitive streak, cells of the notochordal plate will proliferate and eventually it will detach from the rest of the endoderm. So it separates from the rest of the endoderm and now it forms a solid cord of cells, and that is already the definitive notochord. The definitive notochord will underlie the neural tube and will actually serve as the basis for the actual skeleton. Because the elongation of the notochord is actually a dynamic process, the cranial end forms first and the caudal regions are added as the primitive streak would assume a more caudal position. This tells us that structures at the cranial end are actually older as compared to structures found at the caudal end that develop much later in embryonic development. During gastrulation, dorsal and ventral structures that are derived from the mesoderm are established. So why exactly is the heart ventral while the spinal cord is dorsal for a normal individual? This would be because of genes that are responsible for dorsalization and ventralization of the mesoderm. The bone morphogenetic protein 4 or BMP4, which is found throughout the embryonic disc, together with the FGF or fibroblast growth factor, will lead to the ventralization of the mesoderm. Whenever BMP4 and FGF is present, the intermediate mesoderm will become ventralized to contribute to form the kidneys. Meanwhile, the blood, the body wall mesoderm are formed upon the ventralization of the lateral plate mesoderm. Now, because BMP4 is present all throughout the embryonic disc, it means that all of the mesoderm will become ventral if the activity of BMP4 is not blocked by other genes that are expressed in the node. For this reason, the node is called the organizer. Genes, for example, like the chordin, nogin, and follistatin would antagonize the activity of BMP4. As a result, the cranial mesoderm is dorsalized to form the notochord, the somites, and the somatomeres. Later, these three genes would be expressed in the notochord and will become important in neural induction in the cranial region. The antero-posterior axis is signaled by cells at the anterior margin of the embryonic disc. This area, called the AVE, or the anterior visceral endoderm, expresses genes that are essential for head formation. Examples would be transcription factors OTX2, LIM1, HESX1, and the secreted factors Cerberus and LEFT, which would inhibit nodal activity in the cranial end of the embryo. Now, these genes would establish the cranial end of the embryo even before gastrulation. Now, the primitive streak itself is initiated and maintained by the expression of nodal. This nodal gene belongs to a family of transforming growth factor beta or TGF-beta family. Now, once the primitive streak is formed, the nodal would upregulate a number of genes that are responsible for the formation of dorsal and ventral mesoderm and the head and tail structures. Another member of the TGF-beta family, which would be the bone morphogenetic protein 4 or BMP4, which is secreted throughout the embryonic disc, also plays a role in the formation of the antero-posterior axis. The nodal gene is involved in initiating and maintaining the primitive streak. There are also genes found in the node that would later on induce regional specificity in the forebrain and the midbrain areas. In other words, normal formation of the brain relies on the proper distribution or the proper expression of certain genes. An example of that would be the guscoid, which actually activates inhibitors of bone morphogenetic protein 4 and contributes to the regulation of head development. Overexpression or underexpression of the guscoid gene in laboratory animals were found to result to severe malformation of the head region, including duplication of the head. In an experiment involving frog embryos, they were able to find out that if the guscoid gene is overexpressed, it will result into a two-headed tadpole. So in the case of man, such as conjoined twins, they are actually thinking that this might be because of overexpression of the guscoid gene. The regulation of dorsal mesoderm formation in the middle and caudal regions of the embryo is controlled by the Brachiory gene, which is found in the node, the notochord precursor cells, and even in the notochord. What this gene does is it actually regulates the migration of cells in the primitive streak, and it also antagonizes BMP4 in the dicaudal region. Because of the Brachiory gene, cells will form notochords, somatomeres, and somites in the area. However, if Brachiory gene and its protein product is absent, it will result to the shortening of the embryonic axis because there will be no cell migration in the primitive streak. And the shortening of the embryonic axis is what you call as caudal dysgenesis. The degree of shortening depends on the time at which the protein product of the gene becomes deficient. So if it became deficient earlier in development, then you would expect that the embryonic axis will be much shorter. This baby has a severe form of caudal dysgenesis, otherwise known as sirenomelia. Sirenomelia can occur in varying degrees and is caused by abnormalities during gastrulation. So it's also known as caudal agenesis because of the absence of caudal structure, and sometimes called as sacral agenesis because of the absence of the sacral structures. Sirenomelia is characterized by varying degrees of flexion, inversion, lateral rotation, and sometimes fusion of the lower limbs. So when this happens, the baby would resemble that of a mermaid, and so they call it sirenomelia. Most of the time, there could be renal agenesis, imperforated anus, meaning no opening in the anus, and other abnormalities in the genital structures may also happen. So the cause is unknown. It occurs sporadically, but it is most frequently observed among infants of diabetic mothers. So they're actually thinking that caudal dysgenesis may be associated with maternal diabetes in humans. So that's even more reason to be careful of what we eat and what we drink, for instance. Laterality, meaning left versus right-sidedness. Laterality or left and right-sidedness is also established early in development and is because of a cascade of signaling molecules and genes. When the primitive streak is formed, fibroblast growth factor 8 here is secreted by cells in the node and the primitive streak, and it will induce the expression of the nodal gene on the left side, which will help maintain the primitive streak. Other genes that are involved would be the neurotransmitter serotonin. Serotonin is also responsible for further inducing the expression of FGF8. And FGF8 can induce the expression of the nodal and the lefty genes. These nodal and lefty genes are both capable of inducing the expression of the gene PITX2, which is responsible for forming left-sidedness. Now, your serotonin is actually expressed all throughout the embryonic disc. However, because of the presence of the enzyme monoamine oxidase here on the right side, serotonin is metabolized by this enzyme, and therefore you will not find it on the right side of the embryo. Again, if there is an abnormality in the expression of serotonin, it will result to alterations in the positioning of certain organs. For example, it will lead to situs inversus or dextrocardia. By dextrocardia, we mean the heart would be oriented on the right side instead of on the left side. The fate of the cells are also established during gastrulation. So this figure shows the dorsal view of the germ disc showing the primitive streak and also the fate map for the epiblastic cells. Development of embryonic disc and the trophoblast. This is somewhere by the end of the second week of development, a 13-day-old embryo, showing the primary villi of the trophoblastic cells. Remember that the embryo here consists of two layers of cells, you have the epiblastic cells and also the hypoblastic cells. You will also find that the trophoblast is made up of two layers, the cytotrophoblast and the syncytiotrophoblast. During the end of the second week of development, it will also important to note that the primitive uteroplacental circulation is also established. Now, what about the development of the trophoblast during the third week? So, by this time, as mentioned earlier, there's the formation of the three germ layers. And remember, the cephalic end would develop first. Also important would be the formation of the secondary and the tertiary villus, whose capillaries will eventually come in contact with the chorionic plate and also in the connecting stalk, so that you will now form the umbilical cord. And now there will also be an established connection with the maternal endometrium, such that the embryo would be really completely embedded at this time. This figure shows the development of the villus. In figure A, you'll find there the primary villus, which has a core of cytotrophoblastic cells surrounded by syncytiotrophoblastic cells. Eventually, it becomes a secondary villus, whereby the cytotrophoblastic cells are replaced by mesodermal core. And eventually, a tertiary villus is formed when the mesodermal cells develop to become a network of capillaries and venules. So this is a presemitic embryo showing the trophoblast at the end of the third week of development. Here, notice that there's already an extensive secondary and tertiary villi. And there is also the very distinct chorionic cavity here, a connecting stalk, which will become the future umbilical cord. And take a look at this chorionic plate, which surrounds the chorionic cavity here. So this is the maternal endometrium, and these are the blood vessels, maternal blood vessels. Notice how maternal blood vessel will begin to insert itself in the cytotrophoblastic shell that forms the tertiary villi. Notice that the tertiary villi also have already a mesodermal core, and inside would be a network of capillaries as well as venules, which eventually unite to form the connecting stalk, and this is now the umbilical cord. The intervillous space, meaning the space between the villi, is also filled with maternal blood. And therefore, this tells us that there is already a primitive uteroplacental circulation. In fact, the placenta is actually composed of maternal origin as well as fetal origin. And we'll be talking about that later during placentation. So there are tumors associated with gastrulation. If remnants of the primitive streak will persist in the sacrococcygeal region, this will result to a cluster of pluripotent cells that will proliferate or divide to form tumors known as the sacrococcygeal teratoma. And if you are going to open up this teratoma, you will find out that it could consist of mesodermal structures that you will find, for example, say teeth sometimes, hair, other structures might be found there because it's originating from a pluripotent cells. This is actually the most common tumor in newborns, like 1 out of 37,000 newborns. Tumor would be because of the failure of the primitive streak to regress. There is so much to learn about gastrulation, but this is it for now. Thank you for listening. And always remember, life is beautiful.